Since water is the most abundant molecule in the body, anything that concerns water is going to be important. And although water can move in a variety of ways, osmosis is really what operates at the cell level because it's how water moves across a membrane. The way that happens can be a little confusing, so let's explore that in this video. Well, osmosis is another type of membrane transport. But this time, instead of looking at how these particles are moving, we're looking at the other 99+% of the particles there. The water particles, all the blue stuff. So we know that these particles would want to spread out from high to low concentration. We're not concerned about that with osmosis. We're concerned about what happens to the blue stuff. Most of this stuff here. It's going to move in this direction. And understanding why is a little confusing, but let's start with the idea that osmosis is a passive process just like simple and facilitated diffusion. In fact, it's kind of a combination of those two things. So on either side of a membrane you have water, and lots of it. And really the difference between the water concentration on either side is not terribly different. Usually it's these solutes, the stuff dissolved in water, that's more different in a noticeable way. But let's just talk about how water gets across the membrane. It's actually two ways. One, even though water is polar, some of it can sneak through the membrane. Not a ton, but a bit. The cell can also make membrane proteins. And these are called aquaporins. It's a name that makes a lot of sense. Aqua for water, and pores. They're water pores. The more of these aquaporins that the cell makes and puts in the membrane, the less resistance there is to water moving. More aquaporins, more water movement. So cells have some amount of control over how much water comes in and out. So the big question is, what drives the water movement? With other types of passive transport, we talked about diffusion, things moving from a high to a low concentration. But what about with water? I said these concentrations were not terribly different. It's usually the solute concentrations. And there's a lot of stuff dissolved in the watery fluids within our bodies, whether it's inside or outside the cell. There's a lot of different electrolytes and ions. There's proteins, there's sugars. All those are solutes. And so it doesn't matter what it is, but these are going to drive water movement. And kind of a nice rule of thumb is water follows salt. Kind of keep that phrase in mind and don't get too married to the idea that these are salt particles, but any sort of solute, then you got it. So in this case, there are more solute particles over here, which is why I keep drawing water moving that direction. Water is going to move this direction until the solute concentrations on either side balance out. Because eventually there's going to be a lot more water over here, and these particles are going to become diluted until they eventually match the solute concentration over here. That's it. There's a couple different ways I can see this playing out in your mind. One is that water itself moves from a high concentration to a low concentration. Now the difference here is negligible. This is 99.999% water, and this is 99.998% water. But there's still a difference in concentration, so water is going to move down its gradient. The other way, the flip way of looking at this, is from the solute perspective. We could look at water as a helper. Water is trying to move out. Since this solute cannot come across this membrane, water looks at that solute and says, oh, poor guy, let me help you out. Let me move across the membrane so that you can spread out like you want to. And because normally this solute would move over here until concentrations were equal. Well, water can accomplish the same thing. If water moves this way, it will do so until the concentrations of that solute are equal. Whichever way makes the most sense to you is the way I would think about it. Let's look at a couple slides to drive the point home.
All right, so here's kind of an artificial situation with water in a beaker, and it has this semi-permeable membrane running down the middle with different concentrations of solute on either side. And the question is, over here on the right, after some time has elapsed, what's going to happen? Is the water going to move to the right or to the left? Now, you should probably already have guessed that the water is going to move to the right, but let's analyze why. First, let's look at the solute concentrations. On the left, the solute concentration is low, and on the right it's fairly high. The solute wants to move from high to low, but it can't because this membrane blocks it, just like a cell membrane would. Water concentration, on the other hand, is fairly high on the left side and low on the right side, so it's going to want to move to where it's a lower concentration, and it can move through the membrane, so it does, moves to the right. Not a big surprise, but what is kind of interesting is this defies gravity. This is a big enough force that water actually seems to levitate, even though this is a bit of a fictitious scenario, against gravity. And this actually does happen in living organisms. So osmosis is important to generate some force, some water pressure. We can look at that in plants. Here we have 3 plant cells in 3 different situations. Following the principle that water follows salt, if we look at the cell on the left, let's say it's saltier outside the plant cell. Well, water is going to leave the plant cell. It's going to leave it to such an extent, and since these cell walls stay fairly firm here, the rest of the cell shrinks away from it. We call that plasmolysis. This would result in a thoroughly wilted, just flat on the ground sort of plant. Moving to the middle cell, what if it's just as salty outside as it is inside of the cell? Well, then water is going to move out and in equally. Here we end up with a plant cell that's flaccid. It's not super firm. This would be a plant that's kind of leaning over a bit, not quite flat on the ground, but not standing up nice and tall. Moving to the situation on the right, this is what a plant would want. Keep some salt inside, has fairly pure water outside, and so that water is coming into the cell. This actually even needs, he could have flexes the cell wall and makes each cell fairly firm. And all those cells stacked together, we say they're turgid, and they can stand upright.
Well, those examples on the plant, you could see what a difference it made to the cells. And that holds up for the plant as well. So this is a plant, poor thing, that does not have enough water in its cells. Couldn't maintain that pressure in its limp. This one, all that water pressure inside the cells is holding it up, at least mostly. You probably saw some of the terms on there, like hypertonic. So this idea of tonicity is a really tough one, but important even when it comes to us, especially with intravenous fluids. So quick introduction and then a slide. So tonicity always uses the cell as a reference. So it's just basically saying whatever salt level is in here is essentially 0. If we have less salt over here, well, that's a negative, right? It would be less salty than inside the cell, or less solutes. If we have more solutes, well, that would be a positive. And so it's just saying we have more solutes out here. And that then determines where the water goes. Remember, water follows salt. So in this case, there's more salt in here, so the water would come in. In this case, it's going to follow the salts out. Really different results. Let's take a look at that with red blood cells.
Well, here we see red blood cells, kind of like we did the plant cells in 3 different situations. These ones here in the middle are in an isotonic solution and look nice and healthy like a red blood cell should. On the left is hypertonic. That's where there are more solutes outside the cell than inside. The isotonic cells are in solutions with the same amount of salt as in the cell. An example of this that you may have heard of is normal saline. This is a little less than 1% salt solution that you would find in a lot of IV fluids or even things like eye drops. It's a pretty similar saltiness as our body fluids, and so it keeps us healthy. Now, on the right is hypotonic. There are fewer solutes. So let's analyze again what's happening. Big difference here, these are red blood cells. They're animal cells. They don't have a cell wall to protect them. So in the hypertonic case, down here at the bottom, it's really salty outside. Water is leaving the cells and it leaves them shriveled up. Obviously that would not be healthy for us. In the middle, isotonic, salty outside, salty inside, just like it should be. And so water is coming and going and the cells are staying nice and plump, but not too plump. Now let's go to the hypotonic case. It'd be tempting to say, let's give somebody just pure water. That's what hypotonic would be. And so the cells are not any saltier, but they're saltier than the outside of the cell because it's such pure water. In this case, that could even cause the cells to burst. It takes on so much water without a cell wall, it bursts open. Obviously, the two extremes are not good. We want to stay as close to the isotonic situation as possible.
Well, there are a lot of practical applications to osmosis, as you might imagine, since water is so important to life. Not only with plants, by the way, another way that you can get plants to wilt is by over-fertilizing them. Because fertilizer is mostly salt. When you put that in the soil, it sucks water right out. Also, the intravenous fluids. We're bathing our red blood cells in whatever fluid goes in. So it's important that that's isotonic or really close to it. And then a really important one, for people with high blood pressure, they're told to avoid sodium. Sodium is a really big driver of osmosis. Water really seems attracted to salt. And so, people with high blood pressure, if they have too much salt in their bloodstream, that attracts water into those blood vessels, which don't really grow that much, and so it creates a bigger volume, pushing against those vessel walls. That's what high blood pressure is. So there's a good introduction to osmosis. There's a lot more applications we could make with it, and a lot more detail we could go into. I'm sure you'll get into it in the future more. If you keep the idea of water follows salt in mind, then I think you'll usually be able to predict where water goes. An important concept to be sure. Until next time, keep appreciating life, even little things like osmosis.